Good afternoon, everyone. Thank you. I appreciate that. Um, my name is Nikkei Blake, and I have the great honor of serving as Dean of the Graduate Division, Vice Provost of Student Academic Affairs, and I enjoy seeing you all. So welcome to Postdoc Slam. Postdoc Slam happens during Postdoc Association, National Postdoc Association Week. Let me just see for a minute how many postdocs are in the room. Let me just see. Don't hide. That's all right. That's all right. That's all good. All right. I'm gonna come back to you in a second. Eric, I have already messed up your script. Where's Eric? I've already messed up your script. This is just going to happen. What I'm supposed to say is to the very live and lively in-person audience here in Byers Auditorium at our Mission Bay campus, and to those of you who are watching remotely on YouTube, we are glad that you are joining and celebrating with us. As we gather here in person on our campus in San Francisco, we want to acknowledge the Ramaytush Ohlone people who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. All right. Now, Postdoc Slam, as I said before, is part of the celebration of National Postdoc Appreciation Week, a national observance that happens every year in September. So, now, if you're a postdoc, raise your hand. Now we're going to do it. If you're a postdoc, raise your hand. Actually, if you're a postdoc, why don't you just stand up? If you're a postdoc, why don't you just stand up? Stand up if you're a postdoc. All right, all right, all right. Are you feeling appreciated? Don't sit yet, don't sit yet. Are you feeling appreciated? All right, now, if you're sitting next to a postdoc, give them a high five, tap on the shoulder, say, great job, enjoy having you here. Thank you for your hard work. Excellent. All right. Now, Postdoc Slam is first and foremost for the benefit of our postdoc scholars. Today, 10 of our talented UCSF postdoctoral researchers will share their complex and often groundbreaking work in just 3 minutes. Try that at home. All right. This event is not just a showcase of cutting-edge biomedical research, of which you will hear a lot, but it's also an important demonstration of the power of clear and accessible communication in science. At UCSF and around the world, scientists are tackling some of the most vexing health challenges of our time, from pioneering cancer therapies to gene editing technologies to uncovering new treatments for neurodegenerative diseases. Their research holds immense potential to transform healthcare and improve lives. And yet, in today's society, where skepticism towards science and scientists has grown, the ability to communicate the importance of the work that you do has never been more crucial. So, as we listen today, let's celebrate the scientific discoveries and the ability to connect with audiences from all walks of lives, because that is how real change happens. I want to say thank you before they all start. I want them to burn off some energy. So I want to say thank you to our 10 finalists. Why don't you all stand up and let's give you another round of applause. Yeah. Well done. For those of you who are online and not able to hear the vigor in the room right now, there is a lot of excitement in the room right now. It's awesome. Thank you very much. I hope you all give fantastic talks, make the work of the judges really, really hard, and force me to give you multiple prizes. How is that? All right. May the force be with you. Thank you very much. At this time, I am going to get out of the way because I am not your MC. And I would like to turn the program over to our no longer very new Assistant Dean for Postdoctoral Affairs, Dr. Shaquita Allen. Now, don't come up here yet. Hold on. How many of you have met Shaquita? A few of you. All right. If you haven't met Shaquita, before the night is over, you should just go talk to her. She is doing an amazing job for postdocs on this campus, and if you're a postdoc, you should get to know her. Ladies and gentlemen, please help me welcome to the stage Dr. Shaquita Allen.
Thank you. All right. Thank you, Nikkei. I really appreciate that warm welcome. Today, you're going to hear from 10 UCSF postdocs. We have over 1,000 postdocs here at UCSF. These are some of the best and the amazing, talented postdocs that are going to be presented. They've gone through a lot of competitive processes with, I don't know how many, Eric, 60, 70 other postdocs to get to those 10 spots. They've been screened. They were a part of a competitive process. They're going to be reviewed by our judges that are made up of faculty, staff, PIs, and directors of communication and production environments. So we have a lot of experts. We want to thank them again for being here and for assuming this responsibility. They've all been working very hard, our trainees, and of course, it's not just intrinsic motivation, there's some money involved. So $4,000 for the first prize. The second prize is $2,000, and the third prize is $1,000. Plus, there's that People's Choice Award. That's $750. Audience, the remote folks, you'll have a chance to jump in and weigh your opinion there because you'll have a chance to vote, as well as everyone in this room for the People's Choice Award. So that'll be fun, and we'll tell you a little bit more about that in a few minutes. The other way the audience can get involved is the trivia. We'll have some trivia questions in between the presentations. Of course, the people at home can't win a prize because you're there. Just pretend you're playing Jeopardy like you're at home. But people in the room, if you answer a question correctly, you can win a prize. And we have some runners in the room who are going to help you. Sammy will help identify the people. Raise your hand when you want to answer, and then we'll make sure you have access to a prize. Okay? So I know everybody's anxious to hear the talks. The judges, we want to make sure you know their names and know who they are. So I'm going to ask the judges to stand as I introduce each one of them so you know who they are. First of all, Teester Baer Jr. He's the Associate Dean for the College of Sciences and Engineering at San Francisco State University. He's also a professor of chemistry and biochemistry. Dr. Baird is a UCSF postdoc alum and an alum of the judging panel. He was one of our very first Postdoc Slam judges in 2016, and we're happy to have him here. Our second judge is Josh Kornbluth. Josh is a monologist and filmmaker, past Atlantic Fellow for Equity in Brain Health at UCSF Memory and Aging Center, where he launched his most recent project, Citizen Brain. We're excited to hear more about that, too. Our next judge is Wiley Liu. Wiley is the Assistant Vice Chancellor at UCSF, Center for Community Engagement and Anchor Institution Mission within the Office of Community and Government Relations. We have Robin Marks. Robin, please stand. Senior Public Information Rep in UCSF Office of Communications, where she covers new discovery science in various fields. Thank you, Robin. And then we have Ben Palaco. Ben is a computational biologist who serves as lead bioinformatician at UCSF's Quantitative Biosciences Institute, where he works on the Krogan Lab team. Ben is also a UCSF PhD and postdoc alum. So thank you all, judges, very much. Let's give them one more hand. All right. So when you think about the judges, these are the qualities that they're going to be looking for. They're basic qualities in each presentation. The first one is, did the presentation help you to understand the research that was presented? Was the topic and its significance communicated using language that's appropriate to an educated but not specialized audience? And third, did the presentation make you want to know more about the topic they're presenting? Okay? Each talk is going to be limited. That's where the really hard part comes in, as Nikkei mentioned. 3 minutes for each presentation, and so that means we have to time everyone. And if you go over 3 minutes, our presenters will be disqualified. So that is important. And by the way, our official timekeeper is our UCSF registrar, Doug Carlson. Doug is also an official timekeeper for Cal football and basketball. He spots the downs and the yards to go for the scoreboard operator at the San Francisco 49ers. And he also was a clock worker for the NCAA Western Conference Regionals in 2022. So Doug is a real pro. Precision. All right. So finally, housekeeping notes. Please silence your cell phones. Please be respectful of our presenters. Not a lot of whispering, talking, texting. Pay attention to them. They've worked hard for this moment. Let's give them our full attention. So judges, are you ready? Yes. Finalists, are you ready? Okay. Here we go. Here we go. Our first speaker is Roderick Duggar. Roderick's faculty mentors are Margarita Lightfoot and Sherry Lipman. His title is Like PB&J, a complementary approach to reduce food insecurity. Roderick.
When everybody sing it like, yeah, it's good in the hood. I know it is good in the hood.
PB&J, it's the perfect pair. Who doesn't love it? My Nana, that's who. In all my years, I've never seen her eat one. As a child, I was slightly offended by her indifference to my favorite snack. So one day, I had to ask, Nana, why don't you like PB&Js? I'll never forget what she told me. She shared how she grew up in a family with 10 children. For lunch, her parents packed her a PB&J on a biscuit every day from kindergarten through high school. Some days, that was all she ate, which means she did not get enough nutrients. Nana taught me 2 important things that day. First, I learned that food insecurity affects those we would not expect. The USDA defines food insecurity as an inability to get enough nutritious food. Just like my Nana, in 2022, 43% of undergraduate students on UC campuses experience food insecurity. When researchers study this topic, they usually talk about barriers: neighborhoods with no grocery, unaffordable food, and insufficient food assistance. Addressing barriers is important, but a focus on community deficits may hide information about community strengths. Second, I learned that people have a cultural wealth, an array of knowledge, skills, and strategies they use to survive. Like Nana's family, they knew that making PB&J on homemade biscuits was more affordable than purchasing sliced bread. Similarly, young people may use strategies such as meal sharing or buying in bulk to get food. Although people have a cultural wealth, it may be insufficient to prevent food insecurity. So when I heard about a study that gave Black young adults a guaranteed income, $500 per month, no strings attached, I said, sign me up to do research. I always wondered if Nana's story would have been different had her family received a little more income. Her story inspired me to develop 2 research questions about the program's impact. First, I will use quantitative survey data from 300 participants to answer, how does food insecurity change before and after participants receive guaranteed income? And second, through qualitative interviews, I will discover, how does guaranteed income change the way young people apply their cultural wealth to get food? Like the perfect pair of PB&J, my research explores how guaranteed income may complement young people's cultural wealth. The findings will reveal new intervention strategies to build cultural wealth and reduce food insecurity. Thank you.
Awesome job, Roderick. All right, now for our very first trivia question. For those of you who are viewing remotely, again, you can answer amongst yourselves. Our theme today is potpourri, also a little bit like Jeopardy. So this means it's a mixed bag of content that we're going to be covering. A little bit of pop culture, a little bit of science, a little bit of San Francisco, and beyond. So get ready, all right? So raise your hand if you have an answer. Sammy will point to you, and then give us your answer. We have some prizes. We want to thank Campus Life. They've provided a bag that has a little bit of UCSF swag in it, plus a $25 Fast Pay card that's good at some of the campus locations. Okay? And then we're going to tell you a few fun facts around the question after you answer the question. All right. First question. How many gold medals did the U.S. take home in this year's Summer Olympic Games in Paris? Any hands? Guesses? All right, one more guess, and then we'll give it to the closest person.
39, is it?
You're getting close. Very close. Anybody? One last guess? There you go. All right. The USA took first medal count in Paris with 126 total medals. U.S. came in third at the Paralympics with 105 total medals. Closer to home, Amit Elor, Bay Area native, and sibling of UCSF PhD student Ori Elor, took home one of those 40 gold medals in women's wrestling. American ever to win gold in wrestling. And according to NBC, Bay Area Olympians as a whole brought home a total of 68 of the 126 medals from the Paris Games. Rocket, Bay Area. Okay, our next presenter is Rebecca Knoll. Her faculty mentor is Susan Lynch. And the title of her talk is Outcompeting ChatGPT with Poop.
Born this way. 1 in 12 of you were probably born premature. As a pediatrician in Germany, I cared for those tiny, fragile preterm babies. Those babies are at high risk of developing sepsis, a life-threatening infection caused by bacteria. Often, if a baby just seemed a slightly bit unwell, they were given antibiotics to prevent this condition because we doctors are so afraid of it and want to prevent every single case. But this practice leads to antibiotic overuse, health impairments later in life, and most importantly, increases the risk of antimicrobial resistance. So neonatologists worldwide are in need for a better tool to see and predict if the baby in front of them really has sepsis. So what if we could predict this sepsis risk by looking at the baby's poop? Sounds a bit strange, but let me take you into the fascinating world of the gut microbiome and how, together with AI, they could help save lives and prevent sepsis, prevent antibiotic overuse. So our gut is like a bustling city, home to trillions of microbial cells, each with their own genomes. By sequencing those genomes in the baby's poop, we take a census of the city, knowing who is living in the city, what they are capable of doing, and even, if they maybe, are planning a septic attack. Now imagine teaching a super smart AI to become a health detective in the city by teaching it 1,000 of babies' poop samples. So it becomes a health detective and detects danger before it strikes. And here's where I outcompete ChatGPT, because ChatGPT is trained on the poopy data of the internet. So we all got this, right? Please remember to fact check your next conversation with ChatGPT, or you might end up believing the poop that's out there in the internet. Like immigrants eating pets. Yeah. But my AI will be better than this because I spent countless hours in curating my data, improving my algorithms, and fact-checking every single aspect of my research. So that finally, in the future, me and my fellow clinicians have a better tool at hand to detect if the baby in front of us needs really antibiotics, or to say it in the words of Lady Gaga, baby, I was born to survive. Thank you.
All right, thank you, Rebecca. All right, now for our next trivia question. The prominent television and movie star born Alfonso Joseph d'Abruzzo in 1936, founder of the Center for Communicating Science at Stony Brook University in 2009. What is his name? Any guesses? Yeah. Very good. Excellent. Alan Alda earned acclaim and 5 Emmy Awards for his role as Hawkeye Pierce in the classic TV show MASH, where he appeared in all 256 episodes from 1972 to 1983. And he also appeared in dozens of movies from the 1960s until today. But he has also had a long-time commitment to science communication and advocacy, including as host of the PBS show, Scientific American Frontiers, and more recently with his podcasts, Clear and Vivid and Science Clear and Vivid, where his guests have included UC President Michael Drake and UCSF Professor Dr. Edward Chang. The aforementioned Alda Center at Stony Brook is dedicated to empowering scientists to help people engage with complex information through training programs using social science research and improv techniques, all with the goal of making science engaging, alive, and accessible to a broad audience. We'll give the judges a few more minutes. They're still intently taking notes. Close to being ready, judges? Okay, great. Robin, still writing. Okay, our next presenter is Jeremy Ford. Jeremy's faculty mentor is Jean Paz. His title of the talk is Innovating Tools for Driving Breakthroughs in Stroke Pain Research. Jeremy.
While we may not like it, pain is a natural and an important part of our lives. It's actually a good thing that we feel pain to keep us from touching things like hot stoves or walking on injured ankles. But sometimes pain signals can go awry and completely change our lives. So pain is a subjective experience, but a certain type of chronic pain after a stroke has been deemed one of the most debilitating types of pain syndromes. While pain is common in the days following a stroke, central post-stroke pain, or CPSP, develops in up to 35% of stroke sufferers. And it manifests as these feelings of burning, freezing, and lacerating pain that can occur anywhere in the body. Treatments for these patients are typically some combination of pain medications, anti-seizure medications, and antidepressants, but this is sufficient pain management in only about a third of people. Now, the issue is that doctors don't have treatments specifically for CPSP patients, and that's partially because researchers don't have the tools to be able to understand what's going wrong in the brain. So what we do know is that during a stroke, blood flow is limited to part of the brain, resulting in the death of neurons. Now, in CPSP, a common region affected is this deep brain region called the thalamus. So despite the damage from the stroke, the brain has this amazing ability to respond by creating new neural connections and modifying existing brain circuits to be able to preserve the function of the brain. The issue is that somewhere in this rewiring process, something goes wrong in CPSP, creating these persistent pain signals. Now, as I said, the problem is that we don't have treatments specifically for CPSP, and that's partially because the tools that researchers have are limited. And so I'm trying to tackle this problem. My goal is to be able to create a preclinical mouse model of CPSP that will empower researchers to understand better what's going on in the brain. What I'm doing is I'm using a light-sensitive drug and delivering light through a fiber optic directly into the thalamus to create a stroke. I'm currently validating that this model adequately mimics what's going on in CPSP patients. And after this validation, I and other researchers can then apply this model to be able to answer important questions to understand exactly what's driving CPSP. Questions such as, what brain circuits are responsible for this? And are there any molecules that are responsible for driving the interactions resulting in the bad rewiring? And so in this research, I'm hoping to open up the field and allow for breakthroughs so that we can develop treatments specific to CPSP, and in doing so, help the hundreds of thousands of stroke sufferers every year. Thank you.
All right. Thank you, Jeremy. Great job. All right, our next trivia question, to give the judges some time. What is the only bone of the human body that does not articulate directly with any other bone?
Kneecap. Raise your hand, please. Go ahead. Go ahead, in the back. Mandible? Mandible? Yes, in the orange. Kneecap, right. Sorry, what's that?
No.
Yes. Louder?
I can't hear.
What's that? No. Yes.
Yeah, there you go, the winner. The hyoid. The hyoid, also called the lingual or tongue bone, is a horseshoe-shaped bone located in the middle front of the neck, connected to nearby structures via ligaments, muscles, and cartilage. Your hyoid bone is the only floating bone in your body, and the hyoid plays a key role in speaking, swallowing, and breathing. It also provides support for nearby structures like the tongue, the larynx, the epiglottis, and the pharynx, the hyoid bone. Good job. We'll give our judges a couple more seconds. Hang in there. I think they're ready, almost. Teaster? Okay, good. All right, great. Our next speaker is Anna Lyons. Her faculty mentor are Saul Cato and Noel Latoile. Her title is From Microscopic to Iconic: Tardigrades, Neuroscience's New Celebrities. Anna.
Today, we're all here thanks to the incredible interplay of billions of neurons in our bodies. These specialized cells form our nervous systems, allowing us to think, move, and navigate through the world. But studying this vast network is like trying to untangle the world's supply of spaghetti in a tornado. Our brains alone have over 100 trillion connections, roughly equivalent to the total number of stars in the observable universe. So, how can we tackle this complexity in neuroscience? Enter the tardigrade, a tiny transparent animal about the size of a grain of sand. Tardigrades are the smallest animals known to science that have brains, eyes, limbs, and the swagger of a superhero. With only a few hundred neurons, tardigrades can navigate terrain better than any Hollywood stuntman, and they can survive extremes like freezing, drying out, radiation, by shutting down and then reanimating their nervous system as if nothing happened. Only a handful of people study tardigrades worldwide, and as a tardigrade superfan, here at UCSF, I'm developing new methods to edit their DNA to make it so all of their neurons glow only when active by inserting a molecular lantern known as GCAMP. Think of it like there being hundreds of paparazzi in each of the neurons of the tardigrade's bodies that flash only in response to activity. Once I have these glowing tardigrades, I can then put them into teeny tiny mazes or holding devices and use specialized microscopes to track the flashes of light, allowing us to discover new circuits of neurons that underlie behavior and environmental resilience. It's like having a front row seat into a live action movie of a tardigrade's brain. But why should we care how these tiny animals think and move? Tardigrades offer us a rare and unprecedented view into a whole animal's neural circuitry. This would be not possible in a larger, more complex organism, even with the most advanced supercomputer. And by further tweaking the tardigrade's DNA, we can try to mimic diseases like Parkinson's and try to get ideas of how circuits of neurons might underlie movement disorders. And by studying the molecular biology of neural resilience, we could get better ideas of how to protect human brain cells. From microscopic to iconic, tardigrades are making big moves in neuroscience. Thank you.
All right, thank you, Anna. Okay, ready for the next trivia question. It has been suggested that this type of cloud may explain some UFO sightings. What is the name of this type of cloud? Guesses? Anyone? Maybe you need to give them a hint.
Cirrus?
No, not Cirrus. No.
Yes.
Lenticular clouds, that's right. Good work. Awesome. That was a hard one. Very good. So lenticular clouds are dramatic-looking flying saucer-shaped clouds that form mostly in the troposphere, the lowest layer of the Earth's atmosphere. Because lenticular clouds are somewhat rare, many people may have never seen one. Another cloud that forms very low in the atmosphere is one we're more familiar with. In San Francisco. Did you know the fog in San Francisco has a name? Yes, Carl the Fog. You can even follow Carl on Instagram. And he came around this morning pretty much for a while. Awesome. All right, judges. Give the judges a little bit more time, complete their evaluation. Okay, I think they're ready. Our next presenter is Camilla Ting. Her faculty mentor is Jeffrey Bush. And the title of her talk is Zip It, Forming the Upper Lip. Camilla.
Lips like sugar.
remember your first kiss and how your lips felt? And do you remember at the height of the pandemic, we all wore masks and had to smile with our eyes? It was really difficult communicating. We had to shout from 6 feet away, and we couldn't read facial expressions. Was that person smiling at me or frowning? Whether it's for kissing or communicating, our lips play a really big part in our daily lives. And unfortunately, 1 in 700 babies worldwide are born with a cleft lip. Now, we know that certain genes are associated with cleft lip, but we don't know at even the most basic level how these cells and tissue move and take shape to form the lip. So how do we study this? Well, I use a mouse to model this developmental process. And even though we may not look like a mouse, the early stages of lip development are actually quite similar. So in both species, there are these tissue protrusions on the face of the embryo that will fuse to form a continuous upper lip. So I can take a gene that is known to be associated with human cleft lip and remove its function in mice and then just analyze, see how this developmental process goes awry. This is great. I can take tissue sections, I can stain for cell markers, but I really want to know what's happening to these cells in real time during this very dynamic fusion process. But okay, the lips are formed embryonically, so it's not like I could just watch it form like I could watch a tree grow, right? Or can I? In the past, my lab has developed live imaging methods for the mouse embryo. And I thought this would be a great way to visualize the basic processes of lip fusion. So I carefully dissected out the facial tissues from a normal mouse embryo, gave it some warmth, some nutrients, and this dye to label actin, which comprise the skeleton of a cell. This gives the cell support for structure of movement and shape. And then I just took a picture every 20 minutes to make a time-lapse movie. And in this movie, I can see in real time a lot of actin accumulating at the site of where this tissue is fusing. And it appears that the two sides are just zip rings together. So going forward, I like to use this setup to better understand the cellular events in mice that will get cleft lip. And with this information, to infer what's happening in cases of human lip development. So remember, the next time you tell someone to zip their lips, the tissue first zipped up to form your lips. Thank you.
Thank you, Camilla. All right, ready for trivia. Question number 5. This 1960s movie featured UCSF medical students as extras in scenes filmed at what was then called San Francisco General Hospital. What is the name of the film?
Vertigo? What's that?
Vertigo.
No.
Go ahead. Cycle?
No.
Dirty Harry?
It was an action film. Action film. Yeah, there you go. All right, good work. Bullet, starring Steve McQueen, is known for its iconic car chase scene, often hailed as one of the best in cinematic history, and shot on the streets of San Francisco, a must-see classic. The famed car chase spans over 10 minutes of film time and starts under Highway 101 in the Mission and ends near San Bruno Mountain in Brisbane. It's shot over a period of weeks. The chase sequence combined several locations located miles apart and edited together. Bullet. Judges? Okay. Our next presenter is Alexandra Klein. Alexandra's faculty mentor is Mazen Kierbeck. The title of her talk is Take a Deep Breath, Understanding the Neural Basis of How Respiration Influences Emotions. Alexandra.
Every breath you take, every move you make.
I already feel much calmer because, as many of you can relate, I was very nervous just a second ago. Taking a deep breath can also help to calm you down in other situations. For example, when your airplane is taking off, when your experiment has failed, or when you're mad at the Cybertruck driver who just cut you off. The positive effect of breathing on emotions has been known for millennia. However, the underlying mechanism is still unknown. As a neuroscientist, I think the answer is, of course, to be found in the brain, because the brain not only receives information from our outside world, like sights, smells, or sounds, also called exteroception. It also receives information from inside our bodies, including signals from the heart, the gut, and our lungs, also called interoception. So, what happens in the brain of an anxious person taking a few deep breaths? Well, the problem is that the currently available technologies to investigate human brain activity are not precise enough to understand how exactly a breath can impact neural activity. And this is why I'm working with a simpler model organism, the mouse, because a mouse brain is actually very similar to ours, including those brain regions processing anxiety. And to record activity from these brain regions, I can insert a very fine electrode that picks up the electrical signals that neurons use to communicate with each other. And in parallel, I can observe a mouse's breathing. I can then put the mouse into an anxiety-inducing environment, such as a brightly lit box. And because a mouse would probably not reply if I asked how it's currently feeling, I observe its behavior. For example, if a mouse is frozen, like a deer in headlights, it is most likely feeling pretty anxious. Whereas if it is sniffing around and exploring a lot, it is most likely feeling very relaxed. Finally, I can simulate the region in the brain that drives breathing, the so-called pre-Bötzinger complex, to analyze if slowing down breathing can impact neural activity or behavior. Well, does it all work, you may ask now? Well, my preliminary data show that neural activity and breathing indeed fluctuate at the exact same rhythm. And if I slow down a mouse's breathing, I can indeed relieve it from its anxiety. So, what's the point of my research? Well, this research can help develop specific breathing therapies as a non-invasive treatment for a variety of mental health disorders. But for you, it's important to remember, the simplest way how to deal with a Cybertruck driver who cut you off is to take a deep breath. Thank you.
All right, thank you, Alexandra. All right, our next trivia question. Hopefully a lot of you will know the answer to this one. How many electors are in the U.S. Electoral College? Yes, correct. Good word. Civics at work. Each state gets as many electors as it has members of Congress, House, and Senate combined, including Washington, D.C., which has 3 electors. There are currently 538 electors in all. A presidential candidate must receive 270 electoral votes in order to win the presidency. If they receive any less, they will not have secured the majority vote. As a reminder, the election is coming up in November. I encourage all of you to exercise your democratic right and register to vote. Your vote and participation can make an impact on local, state, and national policies. Don't forget to vote. I'll give the judges a few more minutes. Ready? All right, we're ready for our next presenter. Our next presenter is Sonia Nocera. Sonia's faculty mentor is Jonah Chan, and the title of her talk is The Power of a Sugar Mama.
Honey, honey, you are my candy girl. I have to confess, I like mammals. All of them, no discrimination here. I even think humans are cute, especially when they're born. But wait, don't judge me, let me explain. Newborn mammals require just two things to grow strong and healthy: breast milk and sleep. That's it. Breast milk contains all the nutrients to fuel the entire development. How is this even possible? It made me feel like women. We got some power here. I am a neuroscientist, and I study the production of myelin in mice brain. Now, myelin, like the coating of a wire, insulates neuron. Myelinating neurons speed up brain signal up to 100 times, make the brain super efficient. Think of it like the difference between going work, trying to take the super reliable San Francisco public transportation, or zooming there in a Formula One race car. Boom. In particular, for my study, I focus my research on a tiny sugar contained in breast milk. No, it's not any sugar. I like to call it the hero of the brain because it affects the production of myelin in the brain. Look at this slide. I stain in red myelin. These are two slides that come from two mice. One is a normal mouse, and the other is a transgenic mouse. This means that it's genetically modified to not absorb this magical sugar. Notice any difference? It seems that the transgenic mouse have been on a diet. Less red, less myelin. How a tiny sugar can affect so much the entire brain development. It's like to say how a pinch of salt can transform a bland soup into a gourmet dish. Mwah. I want to understand the mechanism underlying this process. I analyze mice brain to figure it out what is the pathway of this sugar and how we can control it. This is crucial, not just for optimize the formula milk, but also for helping people with myelin-related disease, like multiple sclerosis, for example, where myelin is destroyed. And how we can repair, how we can help? Well, often repairing is just the recapitulation of what's happened during development. If we can crack the code, we may be able to produce new treatment to boost myelin production. So what is the take-home message from all of this? Well, everyone deserves a sugar mama, literally.
All right, thank you, Anna. All right, our next trivia question. Are you ready? Scientists think this animal phenomenon involves the larynx, laryngeal muscles, and fibrous pads on the vocal cords, all active during respiration. Or maybe it's just domestic bliss. What are we talking about? Yes, that's correct. The purring of a cat. Cats and their friends are definitely having a pop culture moment. Yeah, we're doing a little purring on screen. But cats have been purring contentedly for eons, and we're still not sure exactly how. Kittens learn to purr a couple of days after birth as part of nursing and bonding with the mother, and continue to purr throughout life, often to show contentment and pleasure, and sometimes to signal injury or pain. Some researchers have suggested the low-frequency vibrations act as a natural healing mechanism, and there's plenty of anecdotal evidence that their purring is healing for human friends as well. Our judges need a little more time. I don't know how many prizes we have. How about are you ready? Okay, they're ready. All right, without delay, our next presenter is Colin Hoy. Colin's faculty mentor is Simon Little, and the title of his talk is Stimulating Positivity. Can a brain implant?
Imagine that you've had depression for years. After fighting for so long, you just want to feel like yourself again, for you and your family. Would you consider a brain implant? Now, we've been implanting pacemakers in brains to treat movement and Parkinson's for decades, and it's starting to work for depression too. The problem is it doesn't work for everybody yet, and that's partly because depression isn't the same for everybody. Some people feel sad, but others feel tired or foggy, and each of those symptoms has a different brain circuit. Now, the brain doesn't care about our labels. The same circuit is driving mood in all of us. And that's regardless of whether your doctor is going to label it depression or bipolar or nothing at all. So here's my pitch. Let's ditch the labels and let's target the specific circuits causing your specific symptoms with brain stimulation. And that's where my research comes in. So up to half of people with Parkinson's end up experiencing depression. And some of those people already have implants for movement, so let's use that to study mood. We asked these people to play special games that are designed to get at the building blocks underneath mood while we're recording their brain activity. And the goal is to find a brain signal that tracks mood fluctuations so that we can use it to control the stimulation like a thermostat for your brain. If the signal's going down, we want to stimulate it, bring mood back up, but not too far. So it turns out, if you want to study mood changes, a really good bet is gambling. And it's not about wins or losses, it's about surprise. Because positive surprise boosts your mood, and negative surprise spoils it. And so in my experiments, patients are flying around hunting for space treasure, and I surprise them with a win, or I blow up their treasure. And I'm actually super pumped because I found a brain signal that's excited for the negative surprise when that treasure explodes, and now I can use that to trigger the stimulation. And the idea here is if we could disrupt that circuit, maybe that negative surprise won't spoil your mood, and we can maintain happy, positive emotions. Now, some of you are probably thinking, wait, you're going to do what with my brain? Is there like a tech billionaire, like some 5G sensors involved here? No, but there's serious questions about who controls and has access to these therapies. And that's why a really important part of my work is about the ethical and societal implications of this research. And in the end, my passion isn't just about improving mental health. It's about thinking about who we are and envisioning who we want to be. Thank you.
Great, thank you, Colin. All right, our next trivia question. This 2012 best-selling book about San Francisco culture features UCSF alum David Smith and the Haight-Ashbury Free Clinic. What is the name of this book? No one? Need a hint? Halloween? Halloween? Got a couple. You have 3 hands. Season of the Witch, but I don't think that's right. That's it? By David Talbot. Great, that was a tough one. Season of the Witch, give her a hand. Season of the Witch. Dr. David Smith, a 1964 graduate of UCSF Medical School, opened the Haight-Ashbury Free Clinic in June of 1967, also known as, nobody wants to bag, also known as the Summer of Love. where it became a haven for anyone in need of medical care, including rock and roll legends like Janis Joplin, Jimi Hendrix, and Jerry Garcia. For decades, the clinic was a pioneer in providing free health care, and Dr. Smith famously declared, healthcare is a right, not a privilege. In the book Season of the Witch, Salon founder David Talbot chronicles the cultural history of San Francisco and from the late 1960s to the early 1980s, from backwater city to thriving metropolis, highlighting such figures as Harvey Milk, Janis Joplin, Jim Jones, and Bill Walsh. Our next presenter is Alyssa Andrew. Her faculty mentor is Nadia Roan, and the title of her talk is Hide and Seek, the game HIV and T cells play.
One way or another, I'm gonna find you. I'm gonna get you one way or another.
How many of y'all remember Tom and Jerry? This cartoon cat named Tom would run around the house looking for his arch nemesis, the mouse, Jerry. And you could feel Tom's frustration every time Jerry got away. Well, I imagine that immune cells in our bodies feel just as frustrated as they roam around looking for our arch nemesis, which are pathogens. Take a pathogen called HIV, for example, or the human immunodeficiency virus. Once HIV enters a cell, a subset of these HIV-infected cells become long-lived reservoirs of HIV that hide throughout the body and are not easily recognized by the immune system. Immune cells, such as T cells, are responsible for killing HIV-infected cells. But in most cases, these cells become overwhelmed and they can't control the virus. Therefore, antiretroviral therapy, or ART, is a treatment used lifelong by people with HIV to help prevent HIV transmission and to control the virus. Interestingly, though, out of the 39 million people living with HIV, about 1% of them do not need treatment to control the virus. These rare individuals are known as elite controllers, and they represent a special case, a rare scenario, where people with HIV do not need treatment to display very long-term drug-free control of the virus. However, we don't understand the complexities of elite control, but we know that the immune system plays an important role. That's why my research investigates T-cell responses in elite controllers and in people who do not need treatment or who do need treatment to control the virus. My goal is to identify novel T-cell signatures that are associated with long-term viral control. To do this, I use a cutting-edge tool known as CyTOF, which acts as a magnifying glass and allows me to look at T-cells in very high detail. By using this approach, I have measured the expression of proteins on the surface and inside of T-cells in elite controllers and compared them to cells with people who need treatment to control the virus. And after analyzing millions of cells, so far I've seen that T cells in elite controllers express very high levels of a protein called CXCR5. This protein is important for helping T cells to travel to parts of the body that are known to harbor the HIV reservoir. So it seems that T cells in elite controllers may be better at finding HIV's hiding places. I'm excited to continue this work because my findings will improve future HIV cure strategies. And one way or another, my finding will ensure that HIV has nowhere to hide. Thank you.
Great, thank you, Alyssa. Our next trivia question. A UCSF fetal cardiology nurse made the news this summer for completing what record-setting physical feat?
She swam to the
Mm-hmm. She swam from the Golden Gate Bridge to the Farallon Islands. That's correct. Give her a hand. This past May, UCSF nurse Amy Applehans-Gubser, an experienced distance swimmer, completed a record-setting 29.7-mile open water swim from the Golden Gate Bridge to the Farallon Islands, the first person to complete the swim in the outbound direction. You've heard of swimming with the sharks. Well, they're definitely a concern. along with bats, jellyfish, frigid water, and, Carl the Frog. Remember Carl? Early in her swim, visibility was severely limited due to the early morning San Francisco fog bank. But Gubser told UCSF writer Eric Brooks, during the 17-hour swim, she found strength in thinking about her patients and how they navigate their challenges. She said, I channeled that thought and used that to propel me from mile to mile. And Amy Applehands Gubser. All right, almost ready. Okay, our next presenter is Taylor Rogers. Taylor's faculty mentor is Joanne Spetz, and the title of her talk is Lift Doula Voices for Birth Equity.
I feel good when there's a million stars in the sky. I feel good when the sun is shining so bright. I feel good.
Everyone who wants to have children deserves to have a high quality, just, and safe labor and delivery. However, that's not the case for everyone. For example, Black birthing individuals, despite their socioeconomic status, have some of the worst prevalence of birth outcomes in the United States. And at America's big age, that is simply unacceptable. Obstetric racism is one of the root causes of these inequities. It's the ways in which clinicians and institutions are abusive and violent towards Black birthing individuals during conception, pregnancy, labor, and postpartum. Thankfully, there is an intervention that has shown to improve health and healthcare experiences. They are non-clinical trained professionals who provide one-on-one educational and spiritual support before, during, and after childbirth. They are birth doulas, and their care has increased the levels of respectful patient care in hospital settings, which is particularly impactful and important for Black birthing individuals. Now, the challenge we are facing in the United States is our limited understanding of the doula workforce. As more and more states are expanding insurance coverage to cover doula care and calling for a larger doula workforce, there is an urgent need for research to understand who the doula workforce is. In my research, I am characterizing the doula workforce and investigating their experiences in the workplace. My work is done with doulas and centers them, their communities, and their clients. I'll start with a series of interviews to learn more about their perceptions of doula training and professional development, the impact of policy and standardization on their work, and finally, their experiences working with and on clinical teams. My intention with this work is to facilitate a work environment in which doulas can thrive while doing their amazing work. So if I'm learning from these interviews, hypothetically speaking, that a doula is being undermined by a clinician, or that they can't live off of the payments provided by insurance companies for their care, I've used these findings to develop and field a survey to understand if these challenges are happening at a broader scale, whether that be state to state or across the national US. So if I'm So with all of these findings, I would develop an intervention to aim to support and retain doulas in the amazing work that they do by addressing all of their needs, whatever they may be. So to ensure that everyone has the birthing experience they deserve, we need to invest in community-oriented research on the doula workforce, who for decades have saved countless Black lives and delivered many healthy, beautiful Black babies. Thank you.
All right, thank you, Taylor. Beautiful artwork, too. All right, our next trivia question. There aren't any more. All right, we want to thank all of our presenters, and we want to invite them all to come back up so we can give them a round of applause. right, thank you. You may be seated. We want to thank all of our presenters for educating and entertaining us. They did an outstanding job. Now, this is also the part where the audience can play a role. We talked about that People's Choice Award, whether you're remote or here. There will be a QR code that will show very momentarily. You'll use that QR code, or if you're remote, there's a link under the video that you'll see there. You can take a minute to think about it. Who's your choice? And we will have voting open for 5 minutes only. You only vote once, 1 vote for each person, and the voting will be open for 5 minutes. Only 1 vote per person. This is an honor system. 1 vote per person. And that winner will be our People's Choice Award winner. And then while the audience is voting, and we're working on that, we're going to be whisking our judges away to a secret location. And they're going to be deliberating with the dean on our winners. And we're going to take some time to go out in the atrium and have some refreshments, the rest of you. And then we're going to invite you back at about 5:25 or so, and we'll announce our winners. Okay? So go have some refreshments, wait for the code, vote for your People's Choice winner, and then we will all come back here to celebrate our winners. Thank you all so much.
All right. I'm going to try to stick to my script. Thank you for hanging around. Anyone like to volunteer to be a judge next year? Yeah, this was not easy, and the judges had a terrific time. All right, before I announce the awardees, I just want to, first of all, congratulate all 10 of our speakers today. Phenomenal job. Regardless of the outcome today, you guys should be extraordinarily proud of the work that you have put in to be able to present today. It is a tough job that you did today. So congratulations to you all. However, we're not in the business of giving trophies to everyone. So there's got to be a dividing line. All right. Now, I'm going to forget to do this, so before, you're going to have to put up with me for a little longer. I need to say some thank yous. Postdoc Slam and this whole NPAL week does not happen without a lot of work and effort by a lot of very special people who reside within the graduate division and beyond. So, first of all, how about a thank you to our MC today, Shaquita. Now, I can't see the rooms, but I don't know who's here because this light is really bright. But if you're a screening judge and you're in the room, can you just wave your hand at me? If you're a screening judge, yes. Thank you very much. Is David still here? He's probably so tired he's gone. So David Blanchard from the OCPD office did a phenomenal job of facilitating the individual coaching sessions for these guys. So thank you, David, even in his absence. Give him a hand because he'll probably watch it on video. There you go. Now, the ETS team, these are the guys with the video thing and doing the screen and all of that. They're the most remarkable guys on earth. So please help me thank them. Also like to give a shout out to our Campus Life Services, who every year gives us prizes. Enjoy those bags that you just got. Thank you. And how was the food? Yeah, don't get used to it. I'm just kidding. The folks in the graduate division and the folks in student academic affairs, who every year helps to really help us to navigate this process. Let's say thank you to them. And then every year I give a big shout out to one particular person. This year I'm expanded my reach. because that food that you just ate, a little young lady named Melissa has just transformed our food game in the graduate division. And Melissa really did a lot of the nuts and bolts of this event. And then Eric, where's Eric? Somewhere, there he is. Eric is the brain of this thing, this whole thing. So please help me thank Eric, Melissa, the communications team, and all the events folk that help to make this possible. All right, I'm done talking. It's time for the fun part. And now, we have 4 awards to present this year. First of all, your award, the People's Choice. And anybody ever did this? Write your notes on a piece of, yes. So there are my notes. There we go. The first place, the People's Award, we didn't choose a person for the People's Award, so I'm just gonna call out Wiley. Wiley, come and do the People's Award. I'm gonna ask our judge to deliver the People's Award. Here we go. And the People's Award goes to...
Oh, so I have to open this.
Yes.
Okay. Can't open it fast enough. I'm under pressure here. Super excited to announce Sonia Nacera won the People's Award.
Stay right there. Wiley. You will give that to her. Yeah. Okay, Sonia, this is not the real cash. This is just telling you how much cash will get deposited into your account by magic of some kind. Okay? All right, congratulations. All right, now you all have to act like you're in like the 49ers stadium and give me a drum roll with your feet because this just doesn't work. So ready? And the 3rd place winner will be presented by Robin. All right, here come the feet.
Excellent.
Okay, so, I forgot. I gotta tell them what happened, right? Yeah. So the judging is really hard, and the judges couldn't distinguish between our 3rd place winners. So I said this, the same thing happened at Grad Slam earlier this year, and it's happening again. There's a secret that you have to keep. Okay? We have 2 3rd place winners. That's good. Which means that I have to come up with an extra $1,000 to give somebody. Please do not tell my business manager this, okay? We're just gonna slide this one under the, okay. All right, and the 3rd place winner goes to, drum rolls please, come on, let me hear.
3rd place. You made this really hard for us, can I just say? I wasn't sure we were even up to making these choices. Okay. 3rd place is tied. Alyssa Andrew and Colin Hoy.
All right, we didn't quite prepare for this, so we only give one the envelope and one the card? I don't know. Rock, paper, scissors. There you go. I love it. There you go. Keep going. Awesome. All right. We're going to ask Josh to come and present the 2nd place winner. There you go. And the second place winner goes to...
The second place winner is a powerful sugar mama. And the people's champion, Sonia Nocera. Yes, yes, back up here. This is a great thing to know.
All right, I neglected to say that Sonia is walking away with a cool $2,000. So, for those of you doing the math. All right, and now, revealing the number one participant for the Postdoc Slam of 2024. Presented to you by Taster. Ladies and gentlemen, and the winner is, drum roll please.
Our first place winner is Alexandra Klein.
Yes, you get to keep that thing. No need to tell you how much money oh, I guess I have to tell the people online. 4,000 big ones is what Alexandra just had. All right. Just want to say a huge thank you again to our judges. Thank you to the audience for being such a responsive audience. Thank you to the competitors who did an amazing job. And you all have a great rest of the evening. Thank you for joining us. I think for the first three winners, they're going to want pictures of all of you. So for the contestant, for the four winners, please come up to the front so we can get pictures of all of you.